Sometimes, when you try to copy notes from a classmate or from your teacher's writings on the boards in a rush, you misread a particular word and write it down misspelled. For instance, instead of writing the word polysaccharide with two C's, you may end up writing with only one. If you are quite unfamiliar with the correct spelling, it is possible that you may misspell the word again during a quiz. Would it not be disappointing if you had lost a point for spelling the answer incorrectly? Such an error can also happen within the genetic sequence of an individual. In some cases of cell division and DNA replication, a gene malfunctions and makes an inaccurate copy of the original genetic code. The resulting error may cause a significant change in the biological structure and function of a specific organism. This phenomenon is known as genetic mutation. Mutation has occurred countless times in the past and has affected a variety of species. Some biologists claim that this has played a role in the evolution of organisms and is partly responsible for the biological diversity of Earth. However, mutations may also have adverse consequences on the functionality of an individual. Remember that in the flow of the central dogma, genetic codes specify a particular type of amino acid used for protein synthesis. If one of the genetic code is altered, it also changes the structure and the function of the protein. Genetic mutation refers to the alteration of the genetic sequence of an organism. The main causes of mutation fall under two categories: errors during cell division and environmental factors. 1. Errors during cell division. During meiosis and meiosis, mutations arise when one section of the gene has been deleted, substituted, translocated, or duplicated abnormally. Sometimes, it occurs when there is an incomplete number of chromosomes, an extra chromosome, or a translocated region of a chromosome. Errors during cell division result in congenital conditions and may be hereditary. Environmental factors. Prolonged exposure to harmful chemicals and radiation, including ultraviolet radiation from the sun and artificial light, may trigger mutations. These factors are called mutagens, which can break the bonds between base pairs or disconnect the sugar-phosphate backbone from the rest of the DNA structure. While DNA may be able to repair itself, there are cases that its structure changes permanently, and certain tissues or organs in the body are being damaged. Types of mutation. Generally, there are two genetic components: genes and chromosomes. Gene mutation is an umbrella term for the changes in a particular gene during DNA replication, while chromosomal mutation refers to alterations in the chromosomes during cell division. Letter A. Gene mutation. When you are cooking, baking, or mixing drinks, you have to make sure that the ingredients are complete, sufficient, and combined well. Otherwise, the quality of the final product is affected. The same thing applies to genes. During DNA replication and protein synthesis, genetic information is passed on several times from a DNA or RNA molecule to another until it is converted into proteins. In some instances, a molecule misinterprets the coding and changes a section of the gene sequence, affecting the proteins produced in the organism. There are two main classifications of gene mutations: point mutation and frameshift mutation. Number 1. Point mutation occurs when a single nitrogenous base in the DNA or RNA structure has been substituted with a different base, such that its complementary base is also altered. For instance, the codon CTT and its complementary GAA, which is the code for the glutamic acid in the normal cells, may be mutated to CAT and its complementary GUA, which translates into the valine amino acid during translation. This particular condition occurs in sickle cell anemia, a genetic disorder in which sickle-shaped red blood cells infiltrate the bloodstream. As you can see, sickle cells mix with normal red blood cells in an individual affected by sickle cell anemia. On the other hand, the genetic mutation that caused the sickle cells has resulted from the substituted base in the codon that codes for the hemoglobin protein. Types of point mutation that may occur in organisms. Letter A. Silent mutation happens when the substitution of a nucleotide base results in the same necessary amino acid and has no adverse effect. Letter B. Missense mutation occurs when a codon has been changed because of a substitute base. The case resulting in sickle cell anemia is an example of missense mutation. Nonsense mutation occurs when a substitute base results in a stop codon and fails to produce the necessary amino acid for the completion of protein synthesis. Number 2. Frame shift mutation happens when one or more nucleotide base may have been deleted or inserted in a single codon. Remember that the tRNA molecule decodes a gene transcript through the 3-letter codons that specify amino acid. When one letter is removed or added, the sequence changes and the molecule may translate the code into a different amino. Frameshift mutation can happen in two ways. Letter A. Insertion occurs when an additional nucleotide base has been inserted in a sequence of codons. To demonstrate this mutation, assume that the genetic sequence is a sentence consisting of 3-letter words. In the sentence below, a base represented by the letter Y has been inserted in the word cat, but the 3-letter spacing of each word remains. Notice that the insertion of the new letter pushed the succeeding letter rightward, resulting in meaningless words. In the case of DNA or RNA sequences, an additional base changes the arrangement of the codons and causes errors in the translation of amino acids. Letter B. Deletion is the opposite of insertion. When one nucleotide base has been removed from the sequence, the codons are rearranged and translated into the wrong amino acids. Consider the example. The removal of letter G from big in the sentence exhibits deletion mutation in genes. As you can notice, when letter G is removed, the next letter occupies its space and rearranges the gene sequence. As a result, the altered codons produce different amino acids. Letter B. Chromosomal mutation. A chromosomal mutation occurs when the number, size, or arrangement of chromosomes in an organism changes. Alterations in chromosomes happen commonly during meiosis, when one or more of the 4 daughter cells bear a gene or chromosomal malfunction. Chromosomal mutation can happen in 4 ways. Number 1. Deletion is caused by a missing segment in 1 or 2 chromosome, result in a significant alteration of genes and expression of traits in an individual. Suppose that a chromosome carries the genes A, B, C, D, E, F, G, H, I, J. When deletion occurs, the gene sequence may result in the following. As you can see, the segment DEF has been removed. Number 2. Translocation happens when fragments of 2 non-homologous chromosomes interchange positions, which means segments of gene sequence are transferred to a different chromosome. Suppose that you have the 2 chromosomes A, B, C, D, E, F, G, H and J, K, L, M, N, O, P, Q, R. If these chromosomes underwent translocation, the result would be the following. Notice the fragments have switched places. Number 3. Duplication happens when a fragment of one chromosome connects itself to the homologous chromosome and repeats a part of its gene sequence. Duplication is exhibited in the following. A segment of the first chromosome has transferred to a homologous chromosome, repeating the section ABCDE. Number 4. Inversion. Mutation occurs when the chromosome contains a segment that has been reinserted in reverse. Suppose that a segment of the chromosome ABCDEFGHI has been inverted. It may result in the following. As you can see, the order of genes in the segment has been reversed. An example of mutation based on an abnormal chromosome count is non-disjunction, which happens when one homologous pair of chromosomes fails to separate properly during the anaphase stage of cell division. In this mutation, both the chromosomes move into the only one of two daughter cells produced, resulting in either an excessive or insufficient chromosome count. The two types of abnormal chromosome count. Monosomy occurs when an individual possesses a missing chromosome from a single pair, meaning that he or she has a total of 45 chromosomes only. Turner syndrome, a chromosomal mutation caused by monosomy, happens only in female individuals. It occurs when a female infant possesses an incomplete or missing X chromosome. Girls with an Turner syndrome often have poorly developed body frame, short height, unusual brown spots in the body, underdeveloped ovaries, and often absent menstruation. The figure shows a karyotype of a female Turner syndrome. Trisomy occurs when an individual has an additional chromosome. Various chromosomal mutation disorders that involve trisomy. Down syndrome, Edwards syndrome, Pantau syndrome, Jacob syndrome, Klinefelter syndrome, and triple X syndrome. Down syndrome or trisomy 21 is one of the most common types of chromosomal mutations in the world, affecting 1 out of 800 infants in the Philippines, according to the Down Syndrome Association of the Philippines INS. This condition arises when an individual is born with a third copy of chromosome 21, causing a number of problems in the development of the individual's physical structure and intellectual capacity. Down syndrome is characterized commonly by distinct facial features consisting of almond shape, upward slanting eyes, and a slightly flattened nose, shorter and smaller stature, poor muscle tone, and a deep crease at the center of the palm. The mutation may also result in certain heart defects, digestive abnormalities, and behavioral issues. Another type of disorder caused by trisomy mutation is Pantau syndrome, or trisomy 13. Individuals born with an extra chromosome added to the 13th chromosome pair commonly experience mental retardation and physical disabilities, such as cleft lip or cleft palate, eye defects, and heart problems because of the severe effects of the syndrome. Babies born with trisomy 13 rarely survive or live longer than 6 months. Edwards syndrome or trisomy 18 is a trisomy disorder in which a person is born with a third copy of the 18th chromosome. The effects of trisomy include numerous organ malformations, a clenched fist, and a severe heart defect. Most infants born with Edwards syndrome do not live longer than 3 months. Moreover, the non-disjunction of chromosomes may occur also in the sex chromosomes, X and Y chromosomes, of an individual. When a person is born with an extra chromosome alongside the 23rd chromosome pair, certain disorders cause numerous physical abnormalities in the individual's body. Although caused by monosomy, Turner syndrome is a disorder that concerns the female X chromosome. Thus, it affects only the female population. An example of the disjunction that occurs only among male individuals is Klinefelter syndrome, a condition that arises when a male infant is born with an additional X chromosome joined with his X and Y chromosome pair. This results in an XXY combination. Carrying an XX chromosome causes some parts of his body to look more feminine in structure, as shown in the figure. Notice that Klinefelter affects testicular development, reducing the size of testicles and lowering the amount of testosterone level. It also results in the development of unusually wide hips and large breasts, decreased muscular composition, and reduced hair growth on the chest and face. Most men with his syndrome are considered slow learners but not mentally challenged. Other types of sex chromosome conditions include Jacob syndrome and the triple X syndrome as opposed to Klein syndrome. Jacob syndrome is characterized by an extra Y chromosome added to the sex chromosomes of a male individual. This usually results in taller stature, excessive acne on the skin, and difficulties with speech and reading. On the other hand, triple X syndrome is characterized by three X chromosomes occurring in females. Girls and women born with an additional X chromosome are commonly taller and thinner than the rest of the female population. They also suffer usually intellectual retardation. Engineering and DNA technology. The study of genetics and the effort to understand further the nature of genetic components are ongoing quests for geneticists and biologists in the 21st century. As a scientific field, genetics has been able to explain how gene expression and mutations occur, but most geneticists today are still investigating and conducting research on how genetic factors may be improved for the benefit of an organism. Genetic engineering or genetic modification refers to a set of techniques by which a segment of DNA information is manipulated or altered. The alteration of a DNA component is performed to improve the organism's characteristics. For instance, it may be applied to enhance the nutritional value of a plant products. Genetic engineers today apply DNA technology to determine how genes control a specific biological trait. Sometimes, they have to extract, copy, or insert a part of the DNA into another organism to produce a hybridized organism. Some techniques that genetic engineers use when applying DNA technology are listed in table. Restriction enzymes used to cleave or cut a fragment of DNA at specific restriction sites. Polymerase chain reaction, a method of applying genes to produce millions of copies of a particular DNA segment that carries a desirable trait. Gene isolation, the process of chemically separating or dividing fragments of DNA. Gene identification, used to identify which isolated DNA fragment is appropriate for a certain purpose. DNA sequencing, the identification of the base pair sequence in a DNA segment. Gene mapping, the identification of a gene's location among the 46 chromosomes of human beings. Recombinant DNA technology, a process of combining DNA segments obtained from two different species to create a new DNA molecule. Vector, a DNA molecule used for transferring a section of DNA into the genes of a host cell. DNA technology is commonly applied in the fields of medicine and agriculture. In medicine, genetic engineering is used to diagnose and treat certain genetic diseases like sickle cell anemia and cystic fibrosis, and to manufacture medicines that boost the secretion of certain hormones, such as the insulin used by diabetic patients. It may also be used to correct some genetic disorders through the process of gene therapy. In agriculture, genetic modification is applied to prolong the shelf of crops and plant products, increase their production, and improve the breeding of animals, increase milk production in cows or eggs from hens.